Okay, so our next speaker comes all the way from the University of Torino in Italy. This is Ida Kokalari, and she's going to talk to us about glucose-derived carbon nanoparticles and promising and safe, effective agents for NIR-induced phototherapy of tumours. There we go. Okay.
Okay. Good afternoon, everybody. Thank you for the opportunity to present this work here at NanoMed. The study is focused on the efficacy and the safety of carbon nanoparticles derived from the degradation of glucose. Phototherapies are a group of therapies which involve the irradiation of target tissue in order to have therapeutic effect. In case of the involvement of nanoparticles, we can have two types of therapeutic effect in the same photoabsorbers. So some nanoparticles are able to absorb the electromagnetic radiation and convert it into heat. This type of effect is called photothermal therapy, which induces the death of tumor cells. In the case of nanoparticles, which are able to absorb electromagnetic energy and generate reactive oxygen species, we are talking about the photodynamic therapy. The advantages of using nanoparticles in phototherapies are the possibility to combine three effects on a single nanoparticle. So some nanoparticles can have photodynamic properties, photothermal properties, and they can also be functionalized with chemotherapeutic agents. And this combination of three therapies could lead to an enhancement of the therapeutic efficacy. Also, some nanoparticles are able to absorb in the near-infrared range. Light in this range possesses a lower phototoxicity, and the tissues are more transparent to light. So the light is able to penetrate deeper into the tissue. And also, using nanoparticles for phototherapies, we are able to target the cancer cells by functionalization of the nanoparticles with targeting agents like antibodies or other molecules which are recognized from the receptors of tumor cells. Carbon nanoparticles are attractive candidates for a variety of biomedical applications since they can combine multiple functions on a single nanoparticles. However, there are some issues about the small-scale production of these nanoparticles, and also which is related to the high cost, to the non-uniform particles production, and also some toxicology issues which limited their biomedical application. Carbon nanoparticles are a relatively new type of carbon nanomaterials. They are easy to prepare with a defined size and can be broadly functionalized without modifying their bulk structure. One of the synthesis routes to obtain these nanoparticles is the hydrothermal carbonization, which is the green synthesis, which use cheap precursors like carbohydrate and also non-toxic solvents like water. So in this study, we use the carbon nanoparticles derived from glucose, from degradation of glucose. Using the hydrothermal carbonization route, we were able to synthesize different type of nanoparticles with different diameters, and we optimized the protocol in order to obtain nanoparticles having size near 100 nanometers, as the uptake of nanoparticles should be higher when the nanoparticles are smaller. So the optimized protocol was used more than 20 times in our lab, and it results in the production of reproducible nanoparticles and with a good yield. So the nanoparticles are perfectly spherical in shape, as can be seen by the ECM images. They are also in the nanosize range, about 150 nanometer as a hydrodynamic diameter, and also with a narrow size distribution. The surface of the nanoparticles is characterized by negatively charged groups. As you can see by the measurement of the zeta potential as a function of pH. The zeta potential increased with increase of pH, which suggested that these groups are acidic groups, mainly carboxylic groups, which confer a high stability in the aqueous media. So the acidic groups were confirmed by titration, and we found that each nanoparticle possesses almost 4,000 acidic groups per nanoparticles, which could be a suitable linkage for a wide range of functionalization. ER spectra of the nanoparticles confirm the presence of hydroxylic and carboxylic groups on the surface. Carbon nanoparticles were analyzed also with thermogravimetric analysis. Their results stable for temperatures up to 200 degrees. And then we can observe a dramatic decrease in the weight, which is related to the decarboxylation of the surface and also to the rearrangement of the bulk structure. The bulk structure was characterized by Raman spectroscopy. The spectra is characteristic of carbonaceous material with these two bands, which are characteristic. The G band is characteristic of the presence of graphitic domain, and the D band is the characteristic of the presence of defects in the graphitic structure. The bulk was also characterized by high resolution TEM. Results confirmed the spherical shape of the nanoparticles as it was observed by SEM, and also a low contrast, which is a characteristic of amorphous carbon. However, by high magnification, we were able to detect some crystalline patches embedded in this amorphous structure. The cytotoxicity was evaluated firstly in macrophages, and we found that nanoparticles do not exhibit toxicity for doses up to 18 microgram per milliliter. And also, we evaluated the interaction of the carbon nanoparticles with the macrophages by TEM, and we found that nanoparticles are efficiently internalized, and also we observed a normal morphology of undamaged macrophages. The cytotoxicity was also evaluated towards epithelial cells, and we found no toxicity even at high doses, and also efficient internalization of the nanoparticles inside the tumor epithelial cells. These results were characterized in absence of irradiation. One of the main obstacles for the translation of inorganic nanoparticles into the clinic is their possible biopersistence in the human body, which could lead to toxicity. In the last decade, several studies report the degradability of the carbon nanomaterial by an oxidative enzyme, which is abundantly expressed in neutrophils. His name is myeloperoxidase, and myeloperoxidase is able to degrade single-wall carbon nanoparticles, graphene, and graphene oxide. The mechanisms of degradation involve the formation of highly potent oxidants like hypochlorous acid in the presence of hydrogen peroxide by the enzyme. So we tested the degradability of our carbon nanoparticles in the presence of the full system. So enzyme, hydrogen peroxide, and chloride ion, which should then be converted in hypochlorous acid. And we found that a significant degradation by the enzyme in 4 days of incubation. In order to confirm that the degradation was due to hypochlorite, we repeated the experiments in the presence of a very diluted solution of hypochlorite, and we found almost total degradation of the nanoparticles in 4 days, which was also confirmed by the SEM images. These findings confirm that the nanoparticles are now biopersistent, and they also could be possibly fastly degraded in vivo. Toxicity of nanoparticles is also linked with their ability to produce reactive oxygen species or to induce production of ROS in cells. And also another mechanism of possible toxicity is also the ability to consume endogenous antioxidants. All of these mechanisms can lead to oxidative stress, which can lead to several cellular component damage and also to cell death. On the other hand, nanoparticles that exhibit antioxidant properties were also reported in the literature, and they can be a promising alternative in the treatment of several inflammatory diseases. So, in order to evaluate the redox property of our nanoparticles, we started with the evaluation of the scavenging ability of these nanoparticles toward the most aggressive reactive oxygen species, the hydroxy radical, which were generated by femtoreaction and were detected by EPR spin trapping spectroscopy. So, in the absence of nanoparticles, we have an intense 4-line EPR spectra, which is characteristic of this adduct. So the adduct between the DMPO and the hydroxy radicals. The experiment was performed also using the same conditions, but in the presence of nanoparticles, and we observed a dramatic decrease on the intensity of the hydroxy radical, which are related to radical scavenging activity of our carbon nanoparticles toward hydroxyl radicals. Some carbon nanoparticles are reported to consume glutathione, so we also tested the ability of our nanoparticles to consume glutathione, one of our endogenous antioxidant species in our cells, and we found that nanoparticles are able to consume the glutathione in cell-free tests. So they could have a pro-oxidant activity toward the reduced glutathione. We moved forward and we evaluated the effect on redox homeostasis of carbon nanoparticles in macrophages by evaluating the level of ROS and also to the nitrite oxide. And we found that nanoparticles did not induce ROS or NO production by macrophages under basal condition. More interesting, we found that the nanoparticles are able to induce dose-dependent inhibition of ROS and NO generation when incubated with activating macrophages. We also tested the interaction between the nanoparticles and the glutathione, in this case in cellular tests, and we found that we don't observe a reduction in the amount of reduced glutathione on cells. This results is in contrast with our previous cell-free tests, but we can explain these results by the fact that the glutathione reductase system is efficient in restoring the reduced glutathione, which was oxidized by the carbon nanoparticles. So all these findings suggest an antioxidant activity of our nanoparticles and the possible clinical application of these nanoparticles in those diseases where an improper macrophage activation is not needed. The photoactivity of nanoparticles was tested in cell-free. Okay. So the photoactivity of nanoparticles was tested in cell-free tests using EPR spectroscopy in order to evaluate the generation of singlet oxygen. So we are talking about photodynamic properties. We found that nanoparticles are able to generate a significant amount of cytotoxic oxygen, even higher as in comparison with the positive control, which was a sample of functionalized fullerene. On the other hand, following NIR irradiation, the nanoparticles are able to increase the temperature of the suspension, which reaches a value, let's say, sufficient for cell death induction. Since nanoparticles are able to produce singlet oxygen and heat upon irradiation, we tested their phototoxicity toward the tumor cells. We use A549 tumor cells. And we checked the toxicity of the cells, of the nanoparticles incubated with the cells, and the toxicity of the laser without nanoparticles. And we then performed the experiment incubating the nanoparticles and irradiating with the near-infrared laser, and we found the change in cell morphology related to cell death in the presence of nanoparticles. We also quantified the stress-induced proteins, like heat shock proteins, and in particular, we found an overexpression of heat shock protein 17 in the case of the samples containing nanoparticles, and irradiated with NIR, which is related to the thermal stress and elevated ROS levels. Finally, the outcome of carbon nanoparticles phototoxicity was also evaluated by the investigation of the DNA damage, and we found that the nanoparticles incubated with cell do not induce DNA damage in absence of irradiation. And on the other hand, nanoparticles enhance the NIR-induced DNA damage following NIR irradiation. To conclude, we were able to produce reproducible nanoparticles with good yield and low cost. They are biocompatible in vitro. They can be degraded by a human enzyme. They have interesting antioxidant properties in absence of photoactivation, and they possesses phototoxicity following near-infrared radiation. Work in progress involves the evaluation of the biodegradability in cells in collaboration with Professor Fadil at the Karolinska Institute, the hemocompatibility and decoration with targeting agent in collaboration with Professor Monopoli and Professor Kenny of the RCSI, and also the in vivo investigation of pro and antioxidant properties in collaboration with the University of Turin. So I would like to thank my colleagues and also Biorima for the financial support, and also you for your attention.
Thank you.